during these early developmental stages, how negative impact it's having on the final seed size, which includes its weight, length, and as well as width. So a lot has been known, explored, and studied regarding the impact of increasing day temperatures, increasing day and night temperatures on the whole. For instance, if the heat stress occurs at the time just before the fertilization happens, that results in the poor seed set because of loss of pollen viability. And if the heat stress occurs at the time just after the fertilization, when the endosperm is transitioning from syncytium to cellularization stage, then there is alteration in this time transitioning timing, which ultimately impacts the final grain yield. And if the heat stress occurs in the later stages of seed development for rice, that has direct negative impact on the grain quality at the whole. So what I wanted to say is, lot has been studied and known about increasing day and night temperature at the whole. Only until recently, we have grabbed the attention for increasing nighttime temperatures. As you can see, that this graph, you can see a more of a reddish trend as compared to the graph on the left, which tells about the temperature increment in the maximum temperature, which is the daytime, and the minimum temperature, which is the nighttime. In particular, if we see US, it's almost the double the increase. So the increase in nighttime temperature is more than the increase in the daytime temperature. And why we are concerned of it is because it's negatively related to the rice yield. As you can see here, that how rice yield is negatively correlated with the increasing nighttime temperatures. One of the reasons being increase in the overall plant respiration response because of the increasing nighttime temperature. And also many studies, they have shown that how increasing nighttime temperature, it alters the source to sink translocation of nitrogen and various other non-structural carbohydrates. So our knowledge about the existing variation about high night temperature tolerance, whether it's from the molecular perspective or the physiological perspective, is comparatively very limited. So in an effort to solve that, we tried to screen the diversity panel in rice for the high night temperature resilience. So we kept in mind that we are taking adequate representation of the geographical diversity as well as the genetic diversity. So the set of population which we used for screening for HNT resilience, it had adequate representation of all the subpopulations which are there in rice. So for the experimental setup, this is how we did, we screened these diverse set of lines for HNT. So they were grown in the controlled greenhouse environment until they started flowering. And once they started flowering, we marked the individual floret and we waited for about 24 hours. And then we moved half of the plants to the HNT greenhouse and half of them, we kept them in the control greenhouse. So the temperature which we used here for our HNT treatment is 30-degree daytime and 28 degrees of nighttime, which is about 5 degrees Celsius higher than the control temperature. So after we moved the plants to the stress greenhouse and the control greenhouse, they were kept there until maturity. And at maturity, we analyzed several grain morphology traits, out of which I will be talking in more detail about seed length, width, and weight. So we had very limited knowledge about underlying loci, which could be adequate contributors towards grain length and other morphological traits in rice for the high night temperature tolerance. So if we just look under the control conditions, so we validated our results from the previous findings that we found the most significant peak for grain length, which is GS3. And it's well known that how this particular gene is an extremely important contributor for controlling the grain length. And then another significant peak for grain width, it was detected at chromosome 5. And this region, seed width 5, it is also been reported for a great controller of grain width in rice. So not to our surprise, these two significant SNPs we also detected under high night temperature conditions. But what was interesting was that Fiei1, which is fertilization independent 1 loci, so there was significant SNP which we detected only under HNT conditions for grain width. And this SNP, it was within the intronic region of rice fertilization independent endosperm. And if we see the allelic distribution, so the genotypes which were having the major allele for this SNP, they were having higher seed width as compared to the genotypes which were having the minor allele, especially under HNT. So what makes Fiei1 more interesting is this particular gene, it's expressed particularly during the seed developmental phases, especially during the early seed developmental stages, and it's not expressed at all in the vegetative phase. And apart from this, it also shows temperature variation, and these are the results from the previous studies done in the lab. So all these parameters, that Fiei1 being specific, Fiei1 showing heat stress response, and apart from that, Fiei1 is an important component, one of the important components of PRC2 complex. And this PRC2 complex, it has been shown that how this complex is, it regulates flowering and seed development, especially in Arabidopsis and also in rice as well. So all these parameters, along with that HNT specific SNP, which we detected for grain width through our GWAS analysis, we thought that Fiei1 can be a promising for further characterizing it and to uncover its role under HNT in rice. So for further validating our results, we just took 4 major and 4 minor allelic accessions, and we did the HNT treatment again and examined their grain length and width. And to our surprise, you can see that how the major allelic genotypes, they are having more seed width, seed size as compared to the minor allelic ones under the HNT conditions. So that raised the question that what could be the probable reason. So the answer was that the major allelic accessions, they were having higher expression of Fiei1 as compared to the minor allelic accessions. And as a result, for validating this hypothesis, we generated the CRISPR-Cas9 knockouts and the overexpression mutants, and we tested them under high night temperature treatment. So not to our response, what we found was that the CRISPR lines, they were having significantly lower grain length as well as grain width when they were imposed, when HNT was imposed. And this was further supplemented with the scanning electron microscopy results for the spikelet hull, which shows that how the Fiei1 CRISPR knockouts, they are having very less size of the layer as compared to the overexpressions. I have not presented the numbers over here, but the knockouts, the size was very small for the cells, whereas there was increase in the number of cells per unit area, which could be the probable reason that the Fiei1 knockouts, they were having smaller size when HNT was imposed. And not only grain length and width, single grain weight was also significantly reduced in the Fiei1 CRISPR knockouts, that due to which we asked the question that what could be the probable reason for reduce in the weight apart from the temperature increment. So that was answered from the scanning electron microscopy results of the cross sections of the mature seeds, which showed that how the CRISPR knockouts, they were having more puffier starch granule structures as compared to the control starch granules, which are well packaged polygonal structures. So not only this, I would switch my gears back to the allelic variation that we found in Fiei1, major alleles having higher expression and the minor alleles having the lower expression. So similarly, we found that the genotypes which were having higher expression, their starch granule structures, they were not at all altered, whereas 3 out of the 4 tested the minor allelic accessions, the starch granules, they were more puffier. So quickly, if I have to summarize my slides, the the very first thing is that the nighttime temperatures, they are increasing at a much faster pace than the daytime temperatures, and that is having more negative impact on the crop yield and productivity. And we did GWAS analysis on about 273 using 273 accessions, and we found out one significant SNP which was specific to high night temperature for grain width. We validated our results with the CRISPR-Cas9 knockouts, which shows reduced grain size as well as grain weight, as well as more puffier starch granules. And not only this, there exists the allelic variation, which we supported with the higher and the lower expression of Fiei in the major and minor allelic accessions, which could be, and to our surprise, we also noticed that the number of accessions which were having minor allele, they were very less in the whole diversity panel, like around 15s or 20s, whereas the major ones, they were like more than hundreds. So that could be one of the reasons that this particular allele, it had gotten the negative selection throughout the breeding period, cycles, due to which we have so less number of these accessions. And that is further supported by the starch granule results that we got, which show that the minor allelic accessions, they have puffier grains than the major allelic ones. And this was just a small piece of the large story. It is not the only contributor. We just found out that, okay, what happened? What's the phenotype? But why it's happening is being further studied, like what are the various other transcriptome level changes happening in the due to Fiei misregulation, which will further make this picture more clear. And not only Fiei, we are further characterizing other novel loci that we identified in this study. With this, I would like to acknowledge my advisor, Dr. Harkamal Walia, for always guiding me and supporting me, and Dr. Puneet Paul and Jasprit Sandhu. These are the students and the postdoc with which we did the GWAS and the high night temperature screening for the whole diversity panel. And then Wheat and Rice Center for Heat Resilience, that has been, we established it by the grant fund NSF, and this is a collaborative effort of University of Nebraska and Kansas State and Arkansas State University. With that, I would like to thank you all, and I'm up for any questions. Thank you.
Thank you very much, Balpreet, for your awesome talk here. We've got time for about 2 questions, so anyone would like to come on up?
That was just fascinating. My question is, when you see these puffier starch granules, do you know, is it more of the same starch, or has it altered the starch that's in those granules?
So that's what a follow-up project is, like what's the exact, whether the myeloplast are changing, whether the packaging, the numbers is changing, that's what the follow-up project is, like what actually is happening. This was the very basic observation that we observed on the mature seeds. Now we are also doing on the developing seeds itself, like when are those changes actually being incorporated, so that why we are seeing it in the mature seeds.
Very nice talk. Do you know if there's any genetic interactions with your gene and other grain size genes?
For this particular project, I checked with the GSC5 and GL7. There was some opposite behavior, like if Fiei is more, those genes were less, and but the more clear picture for your question will be from the transcriptome analysis, which we are right now, we just received the results 2 days back. So that will, I think, be a better way to answer your question.
But what's the effect if you have the large grain allele of both loci together? You get an even larger fruit, or sorry, a larger seed, or do they cancel each other out?
So I don't remember exactly which genes we tested, but they were negatively related with Fiei. So if Fiei1 is more, they were less. If they were less, I don't remember exactly because we tested like a list of around 10 to 15 genes. So that was the observation.
Some great conversation. Thank you. Hopefully we continue these conversations in the hallways later. With that, let's thank Balpreet one more time, and we really appreciate you coming
Thank
today.
you. Thank you.
So we'd also like to present you with a token of appreciation for your time, and thanks for driving all the way up here too. We really appreciate it. All right, so we'll get ready for our